Welcome to this ANT webinar, which is free and open to our member countries in the Asian area. This is Xiaojing Guo from Tsinghua University, China, and I'm also a steering committee member of ANT, which is a network supported by the IAEA. It has been running for 20 years, and we do education, nuclear education in nuclear area, nuclear application. So thank you, Professor David, for giving us this webinar. I would like to give a very brief introduction of Professor Cohen. He has over 260 referred journal publications. He's a senior principal research scientist from the Center for Accelerator Science, Australia's Nuclear Science and Technology Organizations, ANSTO. And he has over 500 conference and report presentations. He is recognized nationally and internationally as a leader in the accelerator science field with a very strong research background in materials science, environmental studies, and life sciences. He has been an international expert in ion beam analysis and accelerator systems for the International Atomic Energy Agency, IAEA, since 1991. He is also a fellow of the Australian Institute of Physics and the Clear Air Society of Australia and New Zealand, and an honorary professor in the Faculty of Engineering at the University of Wollongong. In 2009, he was elected a fellow of the Australian Academy of Technological Sciences and Engineering for his sustained contributions to Australian research. Okay, thank you very much, Professor David. The floor is yours. You can start your webinar now.
Good. Thank you very much for that introduction. And good morning to those in Europe, and good afternoon to the rest of us. Good evening. I'll just share now. We've tested this, so hopefully it works. I'm just.
It's perfect. But
I think I have to swap. I've got two screens, so I'll swap the screens. There, you have the screenshot now. Is that right?
Yes, we can see the full screen mode.
Okay, good. All right. Well, I'll be talking about particle accelerators in research, as has already been mentioned. The overview of my talk today will be a little bit about ion accelerators and what they can do. I'll talk about two techniques of ion beam analysis, IBA, and accelerator mass spectrometry, AMS. And then I'll go thirdly into some applications that we're doing and how these accelerators can contribute to research and adding data in areas such as climate change, fine particle pollution, which is obviously a big issue around the world and in Asia at the moment, and also how these ion beams accelerators can be used for irradiation of electronic components in space and for radiating biological cells. Now, as mentioned at the introduction, I've been with the Centre for Accelerator Science at ANSTO, the Australian Nuclear Science and Technology Organisation, for over 4 decades now. And in that time, I think you can hopefully see my little arrow here. We have 4 accelerators now. We have a 1 million volt accelerator, a 2 million volt accelerator, a 6 million volt accelerator, and a 10 million volt accelerator. So we have quite a broad range of accelerators. And each of these accelerators has a different configuration, and I'll show you here. They basically have the same structure. Let's look at this one down here in the bottom left-hand corner. You have a series of ion sources. You have a tank, which has an accelerator voltage in it. This particular one is a 6 million volt machine. So the tank here is charged up to plus 6 million volts. And then you have several high energy beam lines. And I'll talk about these more in a minute. Here, this one here is an accelerator mass spectrometry beam line. And this one here is an IBA beam line. And we have these end stations at the end here where you perform the experiments. And what I need to show you here is that each of these accelerators, if you look at the 1 million volt accelerator here, the top left-hand corner, it's got a different configuration to the 2 million volt machine here, which has the ion source here, the tank, and the end beam stations here. And the reason for that is each of these machines has a different function and a different functionality. And the functionality depends on the rigidity of the ions that we accelerate in the machine. And the rigidity is defined as ME over Q squared, which is the mass times the energy divided by the charge squared. And depending on whether you want to do ion beam analysis or accelerator mass spectrometry, or whether you want to accelerate hydrogen, or whether you want to accelerate uranium ions, the rigidity of these ions is quite different because their masses are different and their energies are different. So the configuration of each of these machines is for specific applications, which we'll talk about in a minute. And you can see our big machine here, the 10 million volt machine, has a different configuration again. The ion source is on the left here. The acceleration tank that you can see in the picture here is quite large. It's in the middle. And then you have the beam lines here. This is an actide beam line. This is a microprobe beam line. And these other beam lines are for materials analysis. So that's the general configuration of the machines. And each configuration is different depending on the application that you want to do. And I give you here a bit of an idea at the Center for Accelerator Science at ANSTO. To run these 4 machines, we have 37 staff who are experts, ranging from technical experts to research and scientific experts. In any one year, we have over 300 users come in from 37 universities around Australia, and people come in internationally. Industry comes in, and various research organizations come in as well. The 4 accelerators I've described to you now, if you look at them, they have 17 high energy beam lines and 10 ion sources. So that's quite a lot of facilities that we can run. And to feed these, we have 12 sample preparation laboratories. So the whole thing is quite a large concern. It's valued here at around about $60 or $70 million. And it supports a wide range of collaborative research across Australia and internationally. And some of the research I've listed here: climate change, environment, earth sciences, materials science. And I'm going to talk a little bit about space and some of the energy technologies we do. Also fundamental physics, and I'll talk a little bit about some of the techniques, atomic and nuclear physics. And we can also do a little bit of cultural heritage. So that's the Centre for Accelerator Science with its 4 accelerators and its staff. As I mentioned in my introduction, we're going to talk about 2 techniques. The first one I want to tell you a little bit about is accelerator mass spectrometry, AMS. AMS is an isotope dating technique. And on the 4 accelerators that I've shown you, we can accelerate any isotope in the periodic table pretty well, from hydrogen right through to actinides and uranium. I've selected 3 isotopes here to show you with the dating technique how we can span just a few years to millions of years with just these 3 isotopes. Now, carbon-14 is probably a dating technique that everybody's heard of. It's got a half-life of 5,730 years. And you can use that to date anything that's got carbon in it, like charcoal from a fireplace or human remains, anything that has carbon, from present or 100 years out to 10 half-lives we go out. So if you do 10 times 5,700, you get about 60,000 years. So the carbon-14 isotope is good out to 60,000 years. If you look at chlorine-36, One of the big things we have here in Australia, because we're very conscious of water and groundwater and things that we have in groundwater, and you want to look at groundwater, underground water that flows around in artesian basins in Australia, you would use a chlorine 36 isotope. This has a half-life of 301,000 years. And so going out to 10 half-lives takes you out to 3 million years. So you've got from 30,000 to 3 million years for that. And that's very good for groundwater management, geological exploration. If you look at the third one I've got here, it's beryllium-10. It's a cosmogenic isotope. It's got a half-life of 1.4 million years. So we go out 10 half-lives to 14 million years, 500. So with just these three isotopes, we can scan everything from the present out to 14 million years. We can look at climate change and archaeology. We can look at groundwater management, geological exploration, soil erosion, and geomorphology. So that's quite a broad range of different things with just three isotopes. So if you have an accelerator that's got the capability of doing almost any isotope in the periodic table, then you've got a very versatile set of machines. And in fact, for the whole of accelerated mass spectrometry, and I'm not going to talk too much more about it today, the essence of it is that you pick the isotope that's commensurate with the time frame that you want to measure, of the date, the length of the date. We do individual atom counting with this. And this is one of the unique techniques of accelerators. We can count individual atoms. We work on very small samples. AMS can be done on 10 micrograms of sample. And I'll show you a little bit later why that's important, looking at ice cores and measuring the dates down through ice cores. If you've got small samples, you get better timing resolution. We can also measure isotopic ratios. So for carbon-14, we can measure the isotopic ratios of carbon-12, 13, and 14 to typically 1 part in 10 to the 15. 1 part in 10 to the 15 atoms, right? Isotopes. And we can do this with a precision of 0.5%. So if you do 0.5% on these years, like 0.5% at 60,000 years, you're starting to get down to very accurate isotopic dating techniques. And as I've said, the aim is to match the isotope half-life to the time scale that you want to measure. And that is accelerated mass spectrometry. It's been around since the mid-80s, and it's quite a powerful technique. Okay, so now let me tell you about the second technique, which is ion beam analysis techniques, IBA. Now, for ion beam analysis, there are 4 basic techniques that are generally used. There are others, but I'm going to talk about 4 today. And if you look at these 4 here, the first one is PIXI, particle-induced X-ray emission. Second one is PIGI, particle-induced gamma ray emission. The third one is PISA, particle elastic scattering analysis. And the fourth one is Rutherford backscattering, RBS. And each of these is a nuclear technique in itself. If you look at PIXI, when you have an ion from an accelerator comes out, an MeV ion, a proton comes out with 3 MeV, say, or 4 MeV energy, that proton interacts with the electrons in an atom, excites the electrons, knocks out an electron from the K shell or the L shell, another electron falls back into that shell and gives off an X-ray. And that X-ray is characteristic of the element that you've hit. So you get a different energy X-ray if you hit iron and a different energy X-ray if you hit silicon. But you get X-rays. Interaction with the electrons, iron gives you X-rays, PIXI. Now the same process, if the ion comes along and interacts with the nucleus, now remember the nucleus is like a golf ball in the middle of a football field, right? It's quite small. So the probability of hitting this golf ball, this nucleus, is quite small. So the cross sections for gamma interactions are much smaller than the cross sections for X-ray interactions interacting with the electrons. But the principle is much the same. The ion comes in, excites the nucleus gets excited. The binding energies in the nucleus are stronger than the binding energies of electrons around the nucleus, and so you get a higher energy photon, which is a gamma ray. You get a gamma ray given off. The next one is PISA, particle elastic scattering analysis. Now, what that is you have a heavy, it's like billiard ball physics. You have a heavy ion comes into the surface of a material and knocks out a lighter ion. So for instance, you could bring in a helium atom to knock out a hydrogen atom out of the surface. You could do that at glancing angle and knock it out of the surface, or you could have a very thin sample and knock it out the back of the surface. And this is one of the few techniques that's very good for measuring hydrogen, because hydrogen is the lightest element of the periodic table, and therefore it's well separated from other ones. So when you get your spectrum here, you get a peak for hydrogen, whereas carbon and fluorine, for instance, sort of overlap, so you don't get resolving element resolution, but the hydrogen is very good. So PISA is very good for measuring light elements in the matrix. Rutherford backscattering is exactly what it says. You have a light particle comes in, hits a heavy nucleus, and bounces back out at you. And this is very good backscattering technique for measuring carbon, nitrogen, oxygen, fluorine for light elements. So these four techniques here, X-rays are very good for measuring things from aluminium to lead, elements from aluminium to lead. Gamma rays, PIGY, very good for light elements like fluorine, sodium, magnesium, lithium. PISA, as I've said, is very good for hydrogen. And Rutherford backscattering is very good for carbon, nitrogen, and oxygen. So if you look at that, what you have here, these four techniques cover all elements from hydrogen right up to lead in the periodic table. And you can do all these non-destructively with high sensitivity, parts per million sensitivity, in a few minutes, and you can measure very small sample sizes. You can measure samples as small as 10 to the minus 9, a nanogram of a sample. And that is ion beam analysis, very powerful analysis technique. Let's have a look at this a bit closer. These are spectra, 4 spectra from the 4 techniques I just mentioned. We have a code called IBAT, an analysis code, that analyzes these 4 spectra to give you elemental concentrations, basically, from the sample that you've hit with your heavy ions. In this case, these spectra are 2.5 MeV protons, 10 nanoamps of current, 10 by 10 to the minus 9 of an amp for 5 minutes. It's non-destructive. Now, the essence of this is, let's look at the X-ray spectrum. First thing you notice is the Y scale here is 1, 2, 3, 4, 5 decades. It's a log plot, 5 decades. So you go down to 10 parts per million down here. And as I mentioned, each peak here is characteristic of what you hit. So aluminium and silicon, characteristics of soil. Sulfur might come from coal burning if you've got an air pollution sample. Potassium could be biomass burning. And then these metals could be industry. But what you're measuring here is everything from 100% down to 10 parts per million in 5 minutes of running non-destructively from your sample. That's the X-ray. The gamma ray technique is exactly the same. You see the resolution of the peaks here is much better. You get fluorine gamma rays, sodium gamma rays. You get a whole range of low energy gamma rays. Again, the energy of the gamma ray is quite characteristic of what you've hit, and the area under the peak here tells you the concentration, as with the X-rays. The area under the peak is the concentration, and the peak energy is the element. Rutherford backscattering. Again, you've got your light ion coming in, hitting a heavy particle in your target. And here you get an edge, you get carbon, nitrogen, oxygen, fluorine, sodium, aluminium, chlorine. You get edges in your spectrum. And again, this is a thin sample. The area under the peak is the concentration, and the peak identification tells you what you've hit. And I've already explained the PISA, where you have a peak for hydrogen removed from all the other elements. So that's what the spectrum looks like. And you can see their log plots here. So we've got parts per million sensitivity. We're doing this in a few minutes of running. We're doing it with very low currents, so they're non-destructive, and we're using MEV ions. That's the ion beam analysis technique. Okay, so why use accelerators? Just to do a bit of a recap of what I've said so far. With both the techniques AMS and IBA, we have very small beam currents, or we're measuring individual ions or individual atoms, if you like. We can also measure individual photons and individual X-rays. So the sensitivity is really very good. We can do the full range of elements in the periodic table. And we can adjust the ion beams to tailor to suit the situations, as you do with isotopic dating. You pick the isotope with the right half-life to look at the timeframes that you want to measure. So there's three basic techniques. They're very sensitive. I've already mentioned here, but AMS can do isotopic ratios to 1 part in 10 to the 15. You've got 10 to the 23 atoms in a cubic centimetre. Very small samples. We don't need truckloads of samples. We can work with nanograms of sample. And the very short irradiation or times with the ion beams, it's just a few minutes. We don't have days or weeks, and it's basically non-destructive. Okay, so let's go and have a look at some applications now. This is an application of accelerator mass spectrometry. And what I want to show you here is a little bit of the climate change things that we can do. The CO2, if you look down the bottom here, you've got an ice fern, it's called, and in it are a whole load of bubbles of carbon dioxide. And these are trapped in the ice in Antarctica. And if you can extract this carbon dioxide by melting the ice, so you get yourself an ice core, you see over the left here, you've got an ice core. You drill down, and we've got ice cores going down kilometres now. In Australia, we're working on an 800,000 year ice core, and we're looking for a 1 million year ice core to drill into the future. And you take the gas is trapped in these ice cores, and because we can deal with very small samples, we can get very good timing resolution across an ice core. And what I show you here is the greenhouse gas concentrations, and I'll be talking a little bit more about greenhouse gases in climate change in a minute. But if you look at these three greenhouse gases we've got here, methane, carbon dioxide, nitrous oxide. This is a 2,000 year record taken from ice cores. Here's 1000 AD. Here's 1850, which is the start of the Industrial Revolution. And what you see is methane. Let's look at the carbon dioxide here. Everybody knows about CO2, a greenhouse gas. You can see this variation here of solar cycles, a normal variation. And then in 1850, we shot up here. And if you look at carbon dioxide, it increased from a value of about 280 parts per million to 410 parts per million now. And that increase has all happened in the last couple of hundred years. So clearly it's not a natural variation. You get the same thing with methane, which is also a greenhouse gas. It's increased from 700 parts per billion to 1800 parts per billion. And the nitrous oxide has increased from 270 to 330. I give you some examples here. I've already mentioned we've got an 800,000 year ice core. We're going down to a new one's being done in Antarctica, going down to 1 million years, goes down 1 kilometre down into bedrock in Antarctica. And so this will give us an idea of what the climate was like 1 million years ago. We can compare that now with current climates and where we might be going into the future. And approximately 1 tonne of ice contains 20 micrograms of carbon. So you can see if we've got an ice core like this and we can measure 10 micrograms, we only need half a tonne of ice to get the carbon dioxide out to measure it. And we can distinguish between carbon-14 that's modern, which comes from livestock and landfill, and ancient carbon than when we burn fossil fuels like petrol and coal, and what's locked in the permafrost. So we can actually date the carbon and see whether the carbon dioxide is modern or if it's old. So that's an application of AMS in climate change. Okay, I think it's also useful. I'm sure all scientists are quite convinced that climate change is real. And there's enough data around now with the IPCC and various committees and people around the world with data that climate change is real. And I've got 4 things here. I show you here the Arctic and Greenland ice sheets. The red is the Arctic ice sheet, Antarctic ice sheet, sorry. And the Greenland ice sheet is the blue. And what you can see here is the ice mass change in gigatons. And this is only going back to 1990s to the present. And you can see quite clearly that there's been over 5,000 gigatons of ice sheet that's been lost from Queensland. Over here on the right, I had the cherry blossom blooming in Kyoto. This one here goes back to 1400. That's 600 years. And you can see that only in the last, since the 1900s, we've got a very steep drop here, where we used to have it quite clearly around middle of April, and now it's dropped to the beginning of April. So it's come back 2 or 3 weeks in the last 200 years compared to what it had done 600 years before. And you can look at the growing seasons here in Tibet. Same sort of thing again. This is only going back to the 1960s, so we're only going back 40 or 50 years. But you can see in that time we used to go from mid-June, now we're going to the end of May. So these things are starting to change. And again, we've got the grape harvest in France. This again is another 600 years, and you can see that the variability here is quite low. We're looking at mid-September, mid to late September, and now we've dropped right back into August with a nice drop. And again, it's in the last few hundred years. So that sort of backs up the whole temperature type changes and climate change that we all, the scientists, agree. And if you go to the IPCC, International Planet for Climate Change, you look at their report from 2021, the 6th report, and I've highlighted some of the things here that you see. The average global surface temperature was 1.1 degrees higher in 2011-20 than in 1850 to 1900. And in fact, we often talk about whether we're going to go past 1.5 degrees, 1.5 degrees. And I've been talking to people recently, and I think most people who are in the know feel we've already missed the 1.5 degree temperature rise, and we're heading for more than that, unless we do more. And I pointed out to you that the CO2 concentrations are now over 400 parts per million. And that's higher than any time in the last 2 million years. So records are telling us we've got a CO2 level now that's higher than any time in the last 2 million years. And that's all happened since the Industrial Revolution in the 1850s. And again, the concentration of methane is also higher than any time in the last 800,000 years. We've also got temperature rise, global mean sea level increases by 20 centimetres since 1901 to 2018. And climate change here has impacts. It reduces our food security and affects our water security. Here in Australia, we're one of the driest continents on Earth. And so it's very important for us to have water security, particularly in our main cities. And we have water rationing when we have droughts. We had a 7-year drought not long ago, and we were rationing water and drinking water in major cities with millions of people in it. And the total anthropogenic warmings in 2019 since 1750 is now 2.7 watts per meter squared. And I'll show you in the next slide how that's broken down and where accelerators can actually do something here. This one here is the climate change, gases and particles. Now, I don't know whether you realize, but there's been a lot of work in the greenhouse gases. That's up in this region here, carbon dioxide and greenhouse gases, methane, nitrous oxides and helium. If you look at that, that sits on the positive side here. It's positive forcing. And the positive forcing of gases is about 3.8 watts per meter squared. And when the first climate change models came out, they weren't including the particles. And they were predicting temperature rises based on gases that were much larger. We're hearing about 4, 5 and 6 degrees temperature rising. And this wasn't happening. The measurements weren't consistent with this. And people were saying, oh, the models aren't right. You haven't got things going. And sure enough, if you go and look at the aerosols down here, the fine particles, down this, you see that's got negative forcing and it's got a larger error. So we need to get some more data to specify it properly. The error on the greenhouse gases now is quite good. The error on the fine particles and how they operate in this particular climate change thing isn't as well known. But the average cooling because the particles cool, they reflect the sunlight back out, is minus 1.1 watts per centimeter squared. And that's why we end up with this total 2.7. So now with the gases and the particles in these models, we're getting closer to predictions that we can believe and are more robust and more consistent. So the important thing is understanding the aerosols a little better. And it's the accelerator-based ion beam analysis techniques that can characterize fine particle pollution. And if you're in Asia, you'll understand fine particle pollution. Some of the big megacities in Asia have quite significant fine particle pollution problems, not just gases like SOx and NOx and ozone, but they also have fine particles going to big cities in China and India and a lot of Asian cities where you have megacities. And I'll talk about that a little bit in a minute. Well, right now, actually. Let's have a look at this. Urbanization of the globe, right? What I mean by that is as we build bigger and bigger cities and people moving into bigger cities generate more pollution. And as of 2008, more than 50% of the world's population lived in cities. And the world population at the moment is about 7 billion, and we're expected to reach 9.1 billion by 2050. So the urbanization is increasing at a higher rate in developing countries, and particularly through Asia. And I note here at the bottom, over 85% of the growth in the world's population between 2000 and 2024 is expected in urban areas of developing countries. I show you here now, Sydney doesn't have much fine particle pollution, but you can find days when we have bushfires and when things like that where you do. And this is the view of the downtown Sydney across one of our famous bays on a good day. And down here you can see it on a polluted day where we have an inversion layer, and the only thing sticking out at the top of the inversion layer is the center point tower. You can see here. So we do have pollution here in Sydney, and there are consequences of that pollution. I'll show you in a minute. Just to continue with this urbanization leading to pollution, every day 100,000 people globally move into cities, and we have more and more megacities, and a lot of these megacities are in Asia. And this whole business of urbanization requires larger infrastructure, more transport, more industry, and it's putting a big strain on the environment and the resources and generating much more pollution. We have coal-fired power stations, we have them here in Australia, lots more automobiles, we have biomass burning of rubbish and bushfires, we have dust storms, and we also have sea spray components if you live near the coast. So there's a whole range of things that go to your fine particle pollution, natural things, as well as anthropogenic or man-made things. Okay, so why use accelerators to study fine particle pollution? It seems like an odd sort of connection. There are four main reasons of why fine particles are critical. First one is health implications. We define a parameter called PM2.5. That's particulate matter with diameters 2.5 microns. Now that's really small. And they travel, you see over here, deep into the lungs. PM10 or 10 micron diameter particles get stuck in the nasal passages, but the PM2.5 go right down deep into the lungs and the alveoli and actually have direct access to the bloodstream. So if you've got a lead particle or a particle with some toxic element on it that has direct access to the bloodstream, because it's small, then you have health issues. So there are health issues associated with fine particles. The other thing is the physics of fine particles. We're talking about particles with 2.5 micron diameters and less. So let's see, if you take a 1 micron sized particle and you look at how that light is absorbed and scattered, you find that when the diameter of the particle is comparable with the wavelength of the light, you get maximum absorption and scattering. And so the particles of PM2.5 or 1 micron to 2 microns are exactly the same, similar to the wavelength of white light that we see. And so your eyes are actually very good pollution detectors. And that's why you can see the brown haze and the brown bands and inversion layers in the atmosphere when you've got PM2.5 or fine particle pollution in the air. The other thing is that PM 2.5 particles travel very large distances. They go around the globe in 3 and 4 weeks. They don't fall out of the air as readily as coarse particles or PM10 particles. Once they're up into the atmosphere or they're put up there by fires, biomass burning, they stay there for a long time. And I've already shown you the effect of climate change on fine particles. We have to understand fine particles better because they have negative forcing compared with the positive forcing of the greenhouse gases. These two things balance each other. So they're the 4 main reasons to study fine particles. Okay, so what is a fine particle? And what do you get if you take a filter paper and you pull air through that filter paper and trap the material on the filter paper? You measure the mass of that filter paper against the particle diameter on the filter paper. Excuse me. You end up with a bimodal distribution like this, has 2 peaks. This first peak from 2.5 microns down is called the accumulation mode, and it comes mainly from combustion products. You burn sulfur and coal, you get sulfur dioxide, turns to sulfates. You get organics, you get nitrous oxides, you get lead and soot, and a whole range of industries. And that dip here is at around about 2.5 microns. And then if you come up the other side here, you get TSP, total suspended particulates. Anything less than 50 microns in diameter will probably stay in the air for a short while, but they're generated by mechanical processes. Soils, for instance, that are windblown soils that are lifted off the ground by wind, sea salt that comes off the ocean, pollen fragments that come out of pollens, flowers and things, and some local industry generating large particles by mechanical modes. So you have the accumulation mode and the mechanical mode. And the interesting thing here is if you take a particle, a pollen or a grain or some large particle, and you try and grind it up, you can grind it down to a certain size, and after that it coalesces and sticks and you can't grind it anymore. If you take a gas molecule like sulfur dioxide and convert it into ammonium sulfate, it'll get bigger and bigger and bigger, and at some point it won't get any bigger and it'll break up. So you have this natural minimum here of PM 2.5. And it's really there. If you go and measure particle sizes by this technique, you'll really have this dip, 2.5. That's where the PM 2.5 comes from. The PM10, the particulate matter with 10 micron diameter, is a medical thing, which is the particles that you're trapped in the nasal passages in your thing and don't go down into your lungs. And as you can see, that is a combination of some mechanical mode and some accumulation mode. So it's a bit confusing to have a PM10. And you'll find most health and pollution things around the world will not, are getting rid of PM10 as a goal or a standard and doing PM 2.5. That's the general trend. So that's fine particles and what they are. Health implications of fine particles. The mortality rates here. This was a study, 6 city studies done in the 90s by Dockery et al. And what you do here is you go to a place with 10 micrograms per cubic meter annual average, and you normalize the death rate of people dying. So you look at all people who die and you take out all the known sources of death, car accidents, cancer, heart disease, and you're left with a residual that you don't understand, you don't know what it is. And if you plot that residual death rate as you go to more and more polluted cities, you get a linear relationship here. And you can see with cities that have an annual average of 30 micrograms per cubic meter, you've got 30% here, more higher death rate than you had at 10 micrograms per cubic meter. So this graph suggested back in the 90s, and it's pretty well confirmed now today, that 15% increase in the mortality rate for each 10 microgram per cubic meter increase in the fine particle, PM 2.5 concentration. And let me tell you, the health department here in Sydney, and we have what we consider a very unpolluted city, our annual average fine particle mass is about 8 micrograms per cubic meter. And our health department says that a city of Sydney with 5 million people, 606 people a year die prematurely because of our PM2.5 particle pollution. That is twice what we kill on the roads in the road toll. So that's quite a significant number. And if you go to some Asian cities in Hanoi and Delhi and other places where the annual average is off this curve, might be 50 or 60 micrograms per cubic meter, you're going to have quite a significant increase in your death rate. And in fact, I've seen data that people who live in megacities in Asia have their life expectancy reduced by 6 years, their total life expectancy reduced by 6 years. So fine particle pollution is pretty significant. So how do we do this? How do we measure it with accelerators? And I've shown you already we can measure a whole range of elements from hydrogen to lead. So if you can measure aluminium, silicon, calcium, titanium, iron, you can measure the soil content. If you can measure hydrogen, carbon, and sulfur, you can measure the coal burning content. And so what you do is you collect the filter paper here. So you see these two, and you put them in your ion beam and you expose them. And you can see there, we can characterize them up to 30 different elements that we can measure, and we can fingerprint the source by looking at the elements that are associated. So sea spray's got sodium and chlorine in it because it's salt, it's got sulfur, about 8%, it's got bromine content. So if we measure all these elements, we can see they're all associated and correlated, we can say that's sea spray. So instead of looking at elements, we can actually look at individual sources. And again, we can do this on 300 microgram samples. And I've already talked about the 4 techniques. I won't go through this again. I'll just show you here. This is our accelerator. It's a 2 million volt accelerator. Here's our sample for doing air pollution studies up in the top right-hand corner. Again, we had this ion beam interaction. We measured the X-rays, the gamma rays. This is the 2 million volt machine. Protons come down here at 4 or 5% the speed of light, 2 to 3 MeV protons. They interact with the sample in here. We measure these 4 techniques and we get the concentration of all the elements. So what is the average fine particle, PM2.5, composition in Sydney? This is a little bit complicated, this graph, but let's have a look at this. This is 30-year average from 1992 to 2023. On the right-hand side here, we have the PM2.5 mass in red, and this dotted red curve is the thing. So we've come down from 12 or 13 micrograms per cubic meter in the 1990s to less than 8, about 7 micrograms per cubic meter. So we have halved the level here. And you can see a nice peak here in 2009. And that peak corresponds to the composition here, one of these compositions. If you look at the soil here, it's green. And you can see the green one here suddenly peaks up and comes down again. We had dust storms, major dust storms in 2009 and increased our fine particles. So we can start to tie the composition of all of these things on a daily basis that we measure every day throughout the year to various sources. And in fact, if you look at the 30-year average, the mass is 8.6. It's 19 or 20% sulfate. It's 19% black carbon. It's 13, 14% sea salt. It's 18% organics. It's 7.5% soil, and it's 10% nitrates. So that gives you the average composition, which we've done with accelerators. But we can go further than that. Let's have a look at this. Let's have a look at the soil, right? Soil was one of the components that we measured by looking at the oxides of aluminium, silicon, titanium, calcium, and iron. And if you measure each of those things and measure their oxides, you get a soil content. And here I have a picture of a dust storm in the deserts in Australia, nice red soil, you can see what it is. But over in the top right-hand corner here, I have the soil fingerprint plotted from 2001 to 2022. And I've drawn a line here at 0.5 microgram per cubic meter. So there are 166 days with the soil greater than 0.5 microgram above this line. And then the question is, where does that come from? Where does that soil actually come from? Is it all this red dust with dust storms? Let's have a look at it in detail. And the way to do this is you do back trajectories, wind back trajectories, with a program called HighSplit, which is available on the web. It's all free. You can do it for every hour of every day, anywhere on the globe, going back 30 years. quite a useful program. And what I plot here, this is Australia. We have a sampling site here on the east coast at Liverpool in Sydney. And I've done back trajectories for every hour of every day from 2001 to 2022. And I've picked 2 days here. The blue trajectory here is the 4th of September 2002. And you can see there are 24 trajectories there, one for every hour of the day. And every time a trajectory, a back trajectory, goes through one of these boxes, which is desert regions in Australia, you put a little dot. And you can see here on the green one, it's the 8th of May 2019. It doesn't really go through these deserts, it just comes out to the ocean here. Okay. And then you can go back for every hour of every day for 30 years. And each of these boxes here are desert regions. And I've labeled them here, 1 to 14. And you can count up the number of dots in each box, and you can see which desert region contributes to the soil measured in Sydney here, in the Sydney Basin. And the surprising thing is, it's not these desert storm things like I showed you, or these single event days here so much. It's actually 41% of the time, box 15, which is down here, contributes to the soil measured in Sydney. And that turns out to be the Riverina area, which is an agricultural area. So farmers ploughing their fields during windy days produce 40% of the soil measured in Sydney. And if you look at the deserts here, look, 14, 7, 13, 6, all of these, they're less than 1% contributing. Even though we know we get these dust storms on 1 or 2 days in a year, but their total average contribution over the whole year, when looking at every hour of every day, is less than 1%. And the agricultural is much larger, it's 40-odd%. So this is where you use accelerator data, measuring soil, and put that with wind data, you end up with even more useful information as to how you do things. I give you another example here. This is contributions from our coal-fired power stations. And this is a map of the east coast of Australia here. These boxes are the 8 coal-fired power stations in New South Wales, burning 25 million tonnes of coal. And at Richmond here, we have a sampling site. And again, I show you 2 days, the 12th of January 2011 and the 10th of January 2016. 2 back trajectories, blue and brown. And you can see that they intersect with these, what we call East Coast, 6, 7, and 8 things. Again, we look at the secondary sulfate measurement, which we get from sulfur and ammonia. It's ammonium sulfate. So we measure nitrogen, hydrogen, and sulfur. And we put here a line, there's 393 days with the secondary sulfate greater than 2 micrograms. So where do all these days where the sulfate is high come from? And we've got the power stations here. So we go away and we have a look. And what we can see is when these power stations were opened, and we can look at this measured at Richmond, we look at the percentage of back trajectories. And what we find here is that what we call the mid-coast boxes 6, 7, and 8, they're here, 6, 7, and 8. So those boxes there contribute 50% of the secondary sulfate coming into the Richmond site here in the Sydney Basin where people live. 17% come from the Upper Hunter, 1, 2, and 3, these boxes up here. And 11% come from the west, 4 and 5 over here. So what we have, accelerators measuring the secondary sulfate by measuring sulfur, hydrogen, and nitrogen, gives us a daily measure of the secondary sulfate coming into the Sydney Basin at the Richmond site between 1996 and 2024. We then do the back trajectories for every hour of every day from 1996 to 24, and we look at the contribution of each of these boxes, each of these power stations. And we can see that the East Coast power stations contribute a hell of a lot more than the northern ones or the western ones. So again, very useful information. Okay, let's switch track now. Let's go and have a look at what we can do in space and space junk. This is an interesting plot here. In 1957, the Earth looked like this. In 2023, we got a schematic where the Earth looks like this. And this is trying to represent the amount of space junk and satellites currently going around the Earth. And if we look at this, there are 36,000 bits greater than 10 centimeters, and that includes satellites. Okay. There are over 1 million bits, 1 to 10 centimeters, floating around the Earth at the moment. And there's 130 million bits, 1 millimeter to 1 centimeter, orbiting the Earth. And these are going around at high speed, so you don't want to be hit by them. So the whole business there is we're putting more junk up into space. And Australia's getting into the space race now, and everything we put into space has electronics in it, and it's exposed to the radiation in space. So the question is, can we use accelerators to give us a feel for whether our electronics will survive in space or not? Well, the first thing is, let's have a look at what sort of radiation is in space. There's the galactic cosmic rays, and these are charged particles that come from outside the solar system. They have very high energies, up to 10 to the 14 MeV. And there's a lot of elements in there. You've got hydrogen right through to the uranium ions coming through, but there's a steep drop off for anything heavier than iron. So these are galactic cosmic rays that come in and hit the Earth. We then have the solar wind, which is really plasma emitted, millions of tons of hot plasma emitted, mainly protons and helium with electrons, again with MeV energies. And it's only our magnetosphere that protects the Earth. So these things get deflected around the magnetosphere of the Earth. And without that, we would have much higher radiation on Earth than we've got. So the question is, when we put something, astronauts or electronics, up into space, will it survive? And in fact, a lot of these high energy protons that come from galactic cosmic rays or the solar wind, they go dump charge into the electronics, and they might turn a 0 into a 1 or something and change it. And these are called single event effects, SEE. So the question is, can we simulate these single event upsets or single event effects that happen in space? Before I get into that, it's instructive to actually look at the types of doses experienced in space. Now in Sydney, our background level, we all live in a background radiation level, is 1.8 millisieverts per year. That's 1,000 microsieverts per year, or 5 microsieverts per day radiation. So if you go and have a chest X-ray, you'll typically get 330 microsieverts. Okay, that's 60 odd times what you get in a day from the background. If you're an astronaut on the International Space Station, you'll get 400 microsieverts per day. Down here on Earth, we get 5 to 10 microsieverts, depending where you live. They're getting 400 microsieverts a day. And if you go out past the orbit here, where they are, around about 400 kilometers out to 500 or 600 kilometers, you'll get greater than 500 microsieverts a day. If you go and have some sort of bone scan with technetium-99, you'll get 6,000 microsieverts in one dose, and that'll last for 3 or 4 days. This business with astronauts actually is quite interesting, because you probably see that you don't see many young astronauts going into space. They're all in their 40s. And the reason for that is that the NASA and places look at the lifetime dose that you get, 5 microsieverts a day or a couple of millisieverts a year. And they say, how much will you get out in space in 6 months? We don't want to give people too much because we don't want them to end up with cancer. So you find there aren't many female astronauts out there, because females have more sensitive organs than males to radiation. And there aren't many 20 or 25-year-olds out there, because they've got longer to live and would have higher backgrounds. And if you give them too much dose for 6 or 12 months in space, they're likely to get cancer. And if you look down here, I've got the risk of cancer is 100,000 microsieverts. And the whole body lethal dose for a human being is around about 5 sieverts, or 5 million sieverts. And in fact, over here, you can have a look. We're talking about going to Mars. I've drawn a line here. This is millisieverts, dose equivalent in millisieverts. So 100,000 microsieverts is a risk of cancer. So anything above that, you've got a risk of cancer. And this one, orange one here, is a 108-day transit to Mars. And this one here is a 500-day to Mars. And we're talking about going to Mars over 3 years. And to do that, you have to go to the moon for 6 months, where you're right outside the Earth's atmosphere, the ionosphere, magnetosphere. So you've got no protection. And you can see that that's above this level where you've got a whole body risk of cancer, 100,000 microsieverts, these things. So it's pretty risky from a radiation point of view to think about going to Mars for 3 years. Okay, I've already mentioned this, that accelerators can simulate the radiation in space. Use an ion with a known energy and a known flux and a known LET, linear energy transfer. So you use MEV ions, transfers MEV to material that it gets absorbed in. We can do the full range of ions here for space. We can run ion fluxes from a few ions to 10 to the 9 ions or even more per centimeter squared per second. And we can do this linear energy transfer from 1 to 50 MeV centimeter squared per milligram quite easily using the ions from our accelerators. And we have very rapid exposure times from minutes to seconds. And in fact, we can deliver several years of dose in space in just a few minutes of running for ions from these accelerators. The other thing we can do is we can focus our beams down to microns from the accelerators. So we can use them to scan electronic chips with micron resolution, and I'll show you a little bit of that in a minute. Here's our heavy ion microprobe. This is our 10 million volt machine here on the right. The ions come down out of the machine here into these two chambers. This is a vacuum chamber here, and this is an in-air chamber. We focus these with a quadrupole. You see down here, we have a 1 centimeter beam comes into this quadrupole. Quadrupole focuses it down. We have a scanning system on the back so we can scan a 1 micron beam over several millimeters. And then if you take a chip, you decap the encapsulation of that chip. So you got a chip, looks like this. That's a schematic of the chip. You put the ion beam in with 1 micron resolution. You can actually see what part of the chip. Is it the memory? Is it one of the resistors? Is it one of the transistors that's sensitive? You dump charge into that chip while it's operating and see when it works and when it doesn't work. And you can do that with a microprobe with micron resolution in a heavy ion microprobe accelerator like the 10 MeV Antares that we've got at ANSTAR. You can bring the beam out into air. And the reason you want to do that, take it out of vacuum, is that if you're looking at living organisms or human cells or biological cells, you clearly can't put them in a vacuum. So this is a vacuum chamber, and on the back of it, you put an external beam through a thin window, and then you can irradiate material. Vacuum chamber, you bring the beam out through the vacuum chamber into a Perspex box, and then you can irradiate not only electronics, but human cells that won't go into vacuum. And we're very good because we're physicists. We understand ion fluxes to doses, how you convert an ion flux to a dose. And again, as I've mentioned, we can do most ions and isotopes. We can do anything up to 100 MeV energies for ions. So it's a lot of energy to dump into a silicon chip, for instance, or into a biological cell. And we can do ranges of these from nanometers to meters with ion currents from femtoamps to microamps. Is very easy to do. And I've already mentioned spot sizes from microns to centimeters. So this enables us to give doses from micrograys to megagrays. That's 12 orders of magnitude of dose. And remember, 5 gray is typically 10 gray is typically a lethal dose. So mega gray is a very large dose. We do this with accelerators. And I've already shown you here about the microprobe that we have going into a chamber. And you can use this to scan across human cells, extract it through thin windows, and we do biological cells, and we can irradiate them with a range of heavy ions from protons right through carbon up to iron ions or even iodine ions. Here's an example of some biological radiation results where we've taken 2 scans. We've had some human cells in a liquid, and we've scanned across this red area here, and we've scanned across this red area here with our ion beams. And then we stain these things, and we pull them out, and we stain them with a green stain, and we can see where the DNA breaks have been. So wherever you see these green dots, that's where the DNA in that particular nucleus has had a double strand break. And you can see that here. It's green, and then you can blow this area up and you can look at the number of, count the number of single or double strand breaks in individual cells when you irradiate this with micron resolution from a microprobe. And there's obviously other things we can do, which I haven't got time to talk about today because we're coming to the end of the talk. We can test proton heavy ion irradiation effects on solar cells. I mean, obviously all these satellites going to space have solar panels on them. And how long are these solar panels going to last once they get out into this harsh radiation environment in space? And we can do that. And I've already mentioned radiating with microbeams on SRAM and memory chips, and looking at how long memory chips will last in satellites. And again, we can even simulate neutron radiation damage in reactors by using heavy ions and proton knock-on reactions and things like this. So there's quite a broad range of materials testing and radiation testing that we can do with accelerators. So to sum up, hopefully I haven't taken too long, megavolt accelerators produce ion beams, can be used to irradiate, modify or characterize materials. We can focus these beams down to 1 micron, or we can blow them up to a few centimeters. We can run them in air, or we can run them in vacuum. The sample preparation of this is absolutely minimal. We can put very small currents, even individual ions on these targets, femtoamps to microamps. We can give doses to targets from microgray to megagray, and we can dump energy, linear energy transfer with protons and helium and a whole range of heavy ions to simulate space and radiation we see in space. And it has an enormous broad range of applications. I've only talked a little bit about environment and biological and modifications, but there's a lot of other applications of accelerators. So I hope this talk has given you a little bit of a feel for the diversity and things that you can do in the applications of megavolt systems around the world. Thank you very much. Got any questions?
Thank you very much, Mr. David, for this very, very interesting webinar about accelerators. You introduced a very wide use and a varied use of the accelerators. Now we would like to open our question time for our audiences. So if our audiences have questions, you can either input your question in the chat box, or you can also raise your hand and open your microphone to speak. Okay, Professor Cohen, I would like to raise a question about your final introduction about radiology. I mean, you use X-ray, you use the accelerator to do some testing, right?
For irradiating biological cells.
So okay, so it's like taking a photo of the very, very small particles, right?
Oh yeah, with the human cells. Yeah, what we do is we focus our ion beam down to a 1 micron spot size, and then we scan it across here. I'm still sharing. Yeah, we take it and we scan it across the target, and the X-rays that come off the target will enable us to draw maps, elemental maps, which you see here.
国际会. Okay.
And you see these maps that you can see here? You can do them with doses, or you can do them with elements.
Oh, yes, yes.
You can do a silicon map, a calcium map, an iron map, and things like that.
Okay, so what kind of radiation detectors do you use? It must have a very high resolution, I think.
Well, the resolution is determined by the beam size. Let me see if I can show you. Well, here here this one here is a germanium detector, which that's the cryostat. Can you see that? Am I sharing still? So we use germanium detectors, we use silicon detectors, and we use what's called surface barrier detectors. There's a whole range of detectors to pick up X-rays, gamma rays, and scattered particles. And they're all done simultaneously.
Oh, yes. Thank you. Thank you. Yes. Do you have any application of using X-ray to scan things? I mean, to penetrate the object and to see a picture or image of the object to see if there.
Yeah, we don't use X-rays. What we do is we use ion beams. So like XRF, X-ray fluorescence, you use an X-ray to excite X-rays. What we do is we use our ion beams to excite X-rays. So we use the ion beam, a proton beam, goes into the sample several hundred microns, and that excites X-rays which come out of the sample. And
Oh,
so
yes.
we then get a picture of the X-rays that are excited. It's exactly the same as X-ray fluorescence, except instead of exciting the X-rays with X-rays, we excite them with ion beams, protons, helium beams, for instance.
So different energy. Thank you.
Yeah, it's the energy of the proton. The other thing is that the background that you get is called secondary electron Bremsstrahlung background. It comes from secondary electrons. It's much lower for protons because the proton mass is 1836 times more than the electron mass. And so you get much lower backgrounds with proton-induced X-rays than you do with XRF or X-ray-induced X-rays.
Yes, thank you very much. I think we have a hand up. AECS. I'm sorry. Would you like to raise your question?
I think she's
maybe
she's muted, or I think that's muted.
Yes. Can we unmute the audience? Maru, would you like to check?
All right.
Yes,
right.
I have.
Hello, can you hear me?
Yes.
Yes,
right,
yes, now.
Right, hello. Thanks so much for the beautiful lecture. I'm actually a biologist. I'm not a physicist at all, and I don't remember really studying anything about radiobiology throughout my entire bachelor, master's, or PhD. So my question is, if we go back to the slide where you actually showed the biology application of the DNA measurement, maybe the final slide, isn't it?
Is that it? Can you see
right.
that?
So is the DNA study done in... Yes, exactly. Thank you so much. So is the DNA study done in vivo or in vitro? I mean, did you have to isolate the DNA and study it, or did we study it inside the cell? Thank you.
Yeah, what we have here is we have the cells floating in a liquid, and that is put inside the beam and irradiated, and then it's taken out of the beam, and the biologist goes away and stains the cells afterwards. Does that answer your question?
All right. So we didn't actually isolate the DNA.
No, we don't take the DNA out of the cells. This little blow up here is one of the cells, and when you stain it, you can see the DNA breaks here. Each one of these cells you can count is a double or single brand break in.
Well, thank you.
In each of the cells, right? Okay. We have done experiments in the past where we take the DNA out of the nucleus, irradiate it, and then put it back in again afterwards and see if the cell grows in a different way or different things to try and look for radiation resistant cells. But this is all done, it's irradiated and then done offline in the lab.
Thank you for your question. And thank you for your answer, David.
Yep.
Let's see if there's any more questions coming up.
I got one here in the chat. How
Yes.
do you distinguish the source of PM 2.5? As some elements also originate from multiple sources. Do you confirm with other techniques? Yeah, look, unfortunately, I didn't bring any slides here where we could actually look at the fingerprinting. It's a talk in itself to do the fingerprinting. But we can distinguish. It's not like an emissions inventory. This is what you have to get used to. There's what's called a receptor site and an emission site. And if you take a series of pollutants that that are emitted from different sources, they go up into the atmosphere, chemistry goes on and they get mixed and then you breathe them. We do all of our measurements at what's called a receptor site, which is where people actually breathe. So the sampler collects this already mix. And so when you do your fingerprint, you get a mixture of different emission sources in the one fingerprint. For instance, if your sea spray, let me see if I can give you an example here. Yeah. Can you see this? Can you see this here? The slide here. This plot here is the coastline of Australia. This is land to the left, and this is ocean to the right. Okay, and these are back trajectories for 24 hours coming off here. Well, if you look at this out here, these have come off the ocean. Okay, and so in 24 hours, this would come off the ocean. It would pass over these power stations and pick up ammonium sulfate. So it's already got sodium and chlorine in it from sea spray, the sea salt, comes in over here, and we measure it here at Richmond. So that's a receptor site with a mixture of two sources, sea salt and secondary sulfate. Okay? Now there's a program, a technique, a statistical program, if you've got enough data, called PMF, positive matrix factorization. And what that does is it takes your mass and splits it into each of these sources that statistically vary in multidimensional space. And that will separate out the sea spray source and the power station source. And the sea spray source or fingerprint will have sodium, chlorine in it, and sulfur and bromine. And the coal-fired power station source will have organics and sulfur, if you've got sulfur in your coal, and carbon and a few other things. And it will measure it out at this. And unfortunately, I haven't got slides here with fingerprints and stuff that we do for positive matrix factorization. It's another talk in itself. But you can separate out, if you measure every day, as we do here for 30 years, you've got enough data to separate 6, 7, or 8, or in some cases 9 fingerprints of different sources that all come into the one receptor site to make up the total mass. And here, in this situation I've just described, it's very easy to distinguish the sea salt fingerprint from the coal-fired power station fingerprint, both of which are mixed when you measure them at the Richmond site. So we can distinguish these things. We don't need other techniques. We can do it all with the one technique. And the whole idea is you have enough statistics and you measure everything. And all you need, because we measure everything from hydrogen to lead, all we need is 3 or 4 elements that are representative of expected sources. So for instance, at the Richmond site, we know that sea salt's a source. So we have to measure sodium, chlorine, sulfur, and bromine, and that'll give us the sea spray. We want to measure the coal-fired power stations. We have to measure organics, hydrogen, sulfur again, and carbon, and arsenic, and things like that. And as long as we have elements that are representative of the different sources, we span the source space, we can separate them if we measure it for long enough. So complicated, as I said, it's another talk in itself, but we don't need an independent technique. That's one of the advantages of ion beam analysis. We measure over 30, 35 different elements every single day for decades, we can separate out the sources. Because some days they're high, some days they're low, and then there's vice versa. And this variability enables us to statistically extract out the various fingerprints for each of the sources. Very powerful technique called PMF, positive matrix factorization. It was developed in the 1990s and it's used extensively now by EPAs around the world. It's very good. Does that answer the question?
I think we have one more question in the. Can we use IBA for carbon dioxide analysis in air?
Probably not. Because carbon dioxide is a gas and you've got to put gas into a vacuum. That's the first thing. So you'd have to work out some way of getting your ion beam into the carbon dioxide. But why do you want to do that? There's so many better ways to measure CO2 in air than using accelerators. I say quite often the accelerator is extremely powerful. You don't want to use a sledgehammer to crack a nut. If you've got another cheaper technique, and there are many sensors around now to measure CO2 in air, you don't need to use an accelerator or an ion beam technique. And they'd be 1/100th of the cost, 1,000th of the cost.
We have another question in the chat box area, which is, how can this technology be applied in cancer research?
Well, what we're doing at the moment, and I've given you a bit of a feel for it here, is we're looking at dose. I gave you a slide here. And look, I hope these slides will be made available. I don't mind if they're available to people. I can make a PDF of them if they want. Let me just see conditions. Yeah. I mean, you can give various doses to biological cells. You can give doses to a whole range of different things, and you can simulate what people get in a lifetime in a few minutes. So you can use accelerators to simulate dose rates on tissue, on a range of things in the biological area. And basically that's what we're doing. And I think I've explained it here. You can look at the number of ions and doses, the typical doses that you want to get. So for research, if you want to give a few milligrade to somebody or a year's worth of dose to somebody, you can do that, or some part of the body or some organ of the body, you can do that and simulate to see whether it survives. Look at survival rates, look at survival rates of individual cells. You can do a whole range of things like that by giving them different doses of different ions. And that's actually quite useful because there's a range of different ions out in space, for instance, that the astronauts are exposed to because of the galactic cosmic rays and because of the solar wind. They're exposed to X-rays, they're exposed to protons, they're exposed to electrons, they're exposed to a whole range of heavy ions, gamma rays, and you can see what effect they have on various parts of their body. And you can do it with animals as well.
Thank you, Professor Cohen.
Yeah.
I think there is no more questions raised in the chat box. So I would like to show our sincere thanks to your presentation today. It's really wonderful. And thank you. And I would like to also announce our next webinar time, which will be November the 28th. So welcome to join us next time. And we will post, and we have already posted our poster on our social medias like Telegram and yes. So you will find the information. Thank you very much.
Thank
and
you. Pleasure.
have a good day.